is the meaning of R.D.T? Recombinant DNA technology. This word R stand for recombinant, D stand for DNA, T stand for technology. Technology. You know very well. The another name of R.D.T is G.E. This is called genetic. E stand for engineering. So it is the another word which can be used for R.D.T. So simple word in shortcut, recombinant DNA technology is called R.D.T. It is also known by another name genetic engineering. So let me discuss the various tools which are used in R.D.T. Here you can say genetic engineering. There are so many tools. Out of so many tools, the one which can be known as enzymes. Enzymes are used in R.D.T. So many different types of enzymes are used. So out of so many, the most important enzyme is R.E. You know very well what is the full form of R.E? Restriction endonuclease. R stand for restriction, E stand for endonuclease enzymes. Another enzyme which are used is polymerase. The complete name of this enzyme which is used in this chapter, you can say biotechnology, that is called TAQ, DNA polymerase. This is the enzyme which is used, which is very important enzyme, which is used in a technique, the technique which is later to be discussed. That technique is called PCR. The full form of PCR is polymerase chain reaction. P stand for polymerase. The word which is given here. C stand for chain. R stand for reaction. So in this technique we use this enzyme whose name is DNA polymerase. Now question, what is the full form of TAQ? TAQ. Here T stand for thermus. THERMUS. AQ stand for aquaticus. AQUATICUS. This is nothing. It is a name of a bacteria. This bacteria, it can tolerate high range of temperature. Yeah, you can say that this enzyme packed DNA polymerase, which is used in this device, a method, a technique which is called PCR, it can tolerate high range of temperature near about 90 to 94 degree. That's why this word therma, or you know very well, therma and thermos, it indicates the temperature. Simply it indicates the heat. This can be used in this reaction. So these are the things which are used. Another enzyme which are used in this chapter R.D.T, that is called ligase. The complete name is called DNA ligase. DNA ligase. As the name indicates, the function of this enzyme is to link the DNA fragments. That's why this is called ligase. It is also known by another name. In biotechnology, this enzyme is called molecular glue. Molecular glue. Or you know very well what is the role of glue to stick anything. चिपकाने का काम करता है। तो हियर हमें क्या करना है, हमें डीएनए फ्रेगमेंट को आपस में जोड़ना है, ज्वाइन करना है। तो दैट्स व्हाई दिस इज द नेम ऑफ एंजाइम व्हिच इज कॉल्ड डीएनए लाइगेज, बिकॉज हियर वी आर गोइंग टू बी linked to DNA fragment to each other. So these are the some enzymes which are used in R.D.T. Another tools which are used in R.D.T. that is called passenger DNA. Passenger DNA, the DNA which can be moved from one cell to another. How this kind of desired DNA it can be moved? It can be moved with the help of carrier. The carrier which can be work here is called vector. That's why vector in R.D.T. is known as taxi. It is behave like as a taxi because with the help of vector we can transfer a desired gene, a desired piece of DNA from one cell to another, from one bacteria cell to another. So that's why this vector is known as a taxi. There are so many different kind of vector which are used here. The most important is called plasmid. That is called a plasmid. It is behave like as a vector. It is work as a taxi because with the help of plasmid we can transfer a particular piece of DNA, a desired gene from one cell to another. Next, fourth tool which are used in R.D.T. that is called host cell. Because our main target is transfer the desired gene, transfer the piece of DNA so that multiple copies of that DNA is obtained. It is possible with the help of host cell, yeah, host organism. Here in host cell mostly we can use the prokaryotic cell, prokaryotic cell. Yeah, you can say bacterial cell. Bacterial cell they can be used here, and the best example of bacterial cell which is used here is called E. coli. E. coli. The complete name of this bacteria is called Escherichia coli. E stand for Escherichia coli. This is the complete name of this bacteria, which is work as a host cell in which recombinant DNA, you can say plasmid is inserted, so that this bacterial cell with the help of asexual production, which is called binary fission, it can be formed the multiple copies within a few seconds. Near about in 18 to 19 minutes, it can be divided. One bacterial cell with the help of binary fission, it can be formed the two daughter cell. So in a whole day, the many copies of bacterial cell are formed. In short, we can say that we can be obtain the multiple copies of desired piece of gene, yeah, you can say DNA, which is inserted with the help of a carrier. The carrier which carry that particular desired gene or DNA is called plasmid. So that's why vector DNA, yeah, plasmid, it is behave like as a taxi. Like in a taxi, we can be moved from one place to another place. So this is the role of vector here. So these are the some tools which are used in a R.D.T. Next, there are so many another enzymes which are discussed. Mainly there are two enzymes which are used, which are discussed earlier. So let me discuss which one. The first one is called methylase. One enzyme is called methylase. Another enzyme is called R.D.T. Methylase. What is the role of methylase? It helps in addition of methyl group. Addition of a methyl group to the DNA. Then what is the function of RE? RE stand for restriction. Restriction endonuclease. What is the function of these enzymes? The major function of these enzymes, they cut the DNA. Cut the DNA into fragments. in biotechnology, these are also known by the nickname. The nickname which are used for these enzymes is called molecular seizure. Molecular seizure. So one marks question that may be set up, which enzyme in biotechnology is called molecular seizure? The example is called restriction endonuclease enzyme. What are their function? Their major function is to cut the DNA into the small pieces, into the desired fragment, so that we can be obtain desired piece of DNA, we can be obtain desired gene from the total DNA. So these are the function of a two enzyme. These restriction endonuclease enzyme, these are belong to a major category. That major category is known as nuclease. Nuclease. As name indicate, those enzymes which work on nucleic acid, nucleic acid, N stand for the nucleic acid. Or which one is called nucleic acid? Any substance which is found in the cell nucleus, which is acidic in nature, so that is called nucleic acid. In simple word, you can say that nucleic acid is of two types, DNA as well as RNA, and the both are known as genetic material. A major role of these enzymes is work on nucleic acid, they work on DNA. So there are two categories. These are again of two types. The first one is called exonuclease enzyme. One marks question again. Second type is called endonuclease enzyme. As the name indicate, exo means outside. It doesn't mean bilkul outside. We can link this one. Those enzymes which cut the DNA, from the ends. Suppose this is called DNA. 5-2, 3-2, 5-. Those enzymes which start to cut the DNA from the tip, from the end. यहाँ से कट करे. This kind of enzyme is called exonuclease. Opposite to this term, endo means inside. Those enzymes which cut the DNA from inside. Suppose this is the inner portion of DNA. At a particular sequence, they can identify a particular sequence and they can start to make the cut at that particular sequence. That sequence is called nucleotide sequence, that is called recognition sequence. Recognition sequence. Again, very important questions. One mark question. What do you mean by the recognition sequence? A particular sequence of nucleotide base pair which is identified by these enzymes which start to make the cut. This is specific for every restriction endonuclease enzyme. It means it is particular for everyone. It is not all for all one. It is specific for everyone. So that's why these are called restriction endonuclease enzyme. R word that is used. R word means restriction. They can't make cut anywhere. They make the cut at a specific position. They choose the specific sequence. That sequence is called recognition sequence. That's why these enzymes are called restriction endonuclease enzyme. Due to specific nature, they make the cut at a specific position. Because they select the sequence at that particular position and they start to make the DNA inside. That's why these are called endonuclease. So basically these are the two types. These are belong to a category whose name is called nuclease. Okay, very good. Till date, near about more than R.E. is discovered out of